do your own two correlation graphs because you have different variables that I assign. Okay, so that's what it will be. All right, and so tomorrow will be exercise 3. All right, now this is acid base and spectrophotometry. It gives you an introduction to some of the tools that we'll be working with later down the road. And make sure that you turn, at least one of you in your group brings a hard copy of the worksheet. Okay, you need lab coats. I can provide the goggles. Unless you want to use your own goggles, you can bring them. Okay, but otherwise I will provide you with the goggles, and you have to bring your lab coats. Okay, all right. So giving a tour through the cell, and unfortunately I left behind somewhere my clicker, so I have to hang over here. I hate that. I like to walk around, you know. I don't want to have to stand over here pushing buttons. But anyway, I went through some of the endomembrane system, and this is a moving system. There's a lot of traffic that is moving there, and so we have from the endoplasmic reticulum, that's the major manufacturing center of the cell. A lot of stuff goes on there. A lot of things are made there, but they have to be shipped to the right part of the cell. And therefore, transport vesicles, they butt off, butt out, and carry cargo to the cyst side of the Golgi, and that cargo can be mixed, all right? And it is sorted as it passes, that material passes through the Golgi from one layer to the next layer, to the next layer, to the next layer, and then they'll cut off on the trans side and go to wherever the destinations happen to be, which is part of the endomembrane system, or outside, okay? And so some of this traffic can go back to the ER. Some of these butt off as lysosomes, okay? And, okay, so I covered that endomembrane system. We'll see more about that traffic in the next lecture as well. Peroxisomes are not part of the endomembrane system, okay? But they contain proteins, so where do their proteins come from? Bound ribosome or free ribosome? Proteins are made, synthesized on ribosomes. This is not part of the endomembrane system. Free, okay? So these operate in the cytosol, free ribosomes. So the oxidases and the catalases and other proteins that happen to be in the peroxisomes, they're made in the cytosol and they're shipped into the peroxisome. This is also true of proteins that happen to go into the mitochondria and into the chloroplast, okay? Because they're not part of the endomembrane system either, all right? And therefore, only the proteins in general, the proteins that are found either in the lumen or in the membranes or outside of the cell of the endomembrane system, those are synthesized on attached ribosomes, bound ribosomes, okay? All right? So I went through the functions here, peroxisomes, energy converting. I really want to emphasize that these have, these two organelles with the endosymbiotic theory, they have a lot of similarities to bacteria, okay, gram-negative bacteria. In fact, based on RNA sequences, okay, so RNA, okay, I said there's many different types of RNA, and I've mentioned ribosomal RNA, okay? Now, what's different from one RNA to the next is the sequence of bases, and it makes each RNA molecule unique, okay? And we can compare the base sequence of these RNAs across life, because all life has ribosomes, okay? And we can do that to see who is more similar to whom, okay? Their evolutionary relationships. And it just so turns out that chloroplasts are closely related to a type of gram-negative bacterium that is called cyanobacterium. Okay, their ribosomal RNA happen to be very similar. Okay, they share a common ancestor. Doesn't mean that cyanobacteria were the ancestor of the chloroplast, but the chloroplast came from an ancestor that very much shared in common the cyanobacteria. It probably was more like a cyanobacterium than it is the chloroplast today. And for the mitochondria, it seems to be related to a group of bacteria that's called alpha proteobacteria. Okay, and and there's a bunch of other evidence that really supports this. Now, obviously, during evolutionary time, well, first of all, let me back up here. The original characteristics that tied them to bacteria that were first observed is one, that they have two membranes. Okay, that in itself is not so great because the nucleus has two membranes around it too. But in the outermost membrane, it has a special structure called a porin, beta-barrel structure, which we only find in the outer lipopolysaccharide membrane of gram-negative bacteria. Both of these are gram-negative bacteria. There's many other groups of gram-negative bacteria too, right? That's one similarity. So in a chloroplast, which membrane is equivalent to the plasma membrane of a cyanobacterium? It has two membranes, just like a gram-negative bacterium. Which one of those membranes would have been the plasma membrane of the original similar? The inner or the outer? Inner or outer? Inner. Okay. That is homologous to the plasma membrane of cyanobacterium. All right. The outer membrane is homologous to the lipopolysaccharide layer, because that is the one that has a big barrel. Okay, that's the one that has porins in it. Okay. Other similarities is that they have their own DNA, and the DNA is a naked circle of DNA. Okay. Obviously, over evolutionary time, a lot of the genes that were originally there have been shipped to the nucleus. Now, chloroplasts have a bigger genome. They can make a lot of their own proteins, whereas mitochondria, they can only make very, very few. Out of the 480 plus proteins that are necessary for the construction of the mitochondrion, most of that is made on free ribosomes and shipped in. Okay. Now, they have their own information for making just a few of those proteins and also transfer RNAs and ribosome RNAs. They have their own ribosomes, and those ribosomes are more similar to ribosomes of bacteria. They're sensitive to streptomycin and tetracycline, and they're small, unlike the ribosomes in the nucleus, or excuse me, in the cytoplasm of the eukaryotic cells. So those are the similarities, but there's much, much more. Okay? All right. Went over that, and I think that was my last slide, right? Okay, so to give you a little bit of comparison here about how much these organelles take up as far as space and number within the cell, there is, and this is based on sort of liver cells in the human body, so it's not, and it's average. So as far as cytosol, there's one unified cytosol. That area constitutes about, in typical liver cells in humans, about 54%. Mitochondria, a typical cell would have 1,700 mitochondria and occupying about 22%. Now, this really depends on what that cell is doing. If this is muscle cells, skeletal muscle cells, the number is going to be much higher, okay? Because muscle cells are using a lot of ATP compared to adipose cells, fat cells, okay? Fat cells would have something lower than that, all right? So these would have to be adjusted according to whatever is going on. Ruppy R, okay, about 9. Smoothie R, Golgi altogether, occupying about 6% and so forth. And you don't have to know any of these numbers, okay? You don't have to know any of these numbers. This just sort of gives you some idea as to what the contents of typical liver cells have to be, okay? Okay, other structures that are found in eukaryotic cells, something called a proteasome, okay? Proteasomes are not membrane bound, right? There's no membranes here, no phospholipid bilayer. They're multimeric, which means that there's a lot of different proteins there. And I'm not going to go into how many there are. This would be more characterized, instead of as an organelle, as a supramolecular system, okay, somewhere between there. But it's a very important thing. It also, like chaperone proteins, have a sort of a trash can look to them. They're hollow on the inside, there's a lumen, they have a lid, and a bunch of different proteins there. And its main function is the degradation of misfolded proteins that, or other proteins that don't need to have them anymore. Remember homeostasis. The cell has to adjust to physiological changes. And so there's times when they have to have a certain protein. That protein fulfills a certain function, but things change. And when they change, we don't need that protein anymore. So we need to recycle it. And so this is for breaking them down. And so these proteins have to be tagged, okay, in order for the cell to know what is wrong with them. And there's a globular protein that's called ubiquitin. Okay, this is a protein that is, it has several functions, not just only for tagging proteins that have to be destroyed, but we have here a protein that's misfolded, or it's old, or we don't need it anymore. It's recognized by a group of other proteins, we're not going to mention them, and a string of ubiquitin units are attached to it, okay, in a linear fashion, right? Several of them. And this is a tag that tells the cellular machinery to destroy this protein. And so it's transported to a proteasome, stuck, lid comes up, stuck down the middle, and a lot of these are hydrolytic. And so they break and chew up the polypeptide, and it's recycled, okay? The parts are recycled. So this is for recycling, all right? Now, for proteins that are in the endomembrane system, they're destroyed by lysosomes when they're old and we have to get rid of them, okay? So if they're in the lumen of endomembrane system, or in the membranes of the endomembrane system, then, or if they are proteins that are taken in from the outside of the cell, they're taken into, they fuse with a lysosome, and those are destroyed. Now, once again, this is just a general, broad generalization. There's many exceptions to it as well, all right? All right, so proteasomes. These are also called heat shock proteins. So if you come across it in your book, heat shock proteins, because they were first discovered in bacteria, Escherichia coli, when they put them through something called heat shock. That's changing their temperature real quickly. First of all, bring it up to 40 degrees, then back down to 35 or something like that. And that is a stress on the bacterium. That change in temperature probably causes a lot of misfolded proteins. Okay, your proteins, you have to keep a constant physiological condition. Something I failed to mention when I went over protein structure and denaturation is if you change a physiological condition such as temperature, it can lead to misfolding of proteins, denaturation. Why is that? Why would an increase in temperature affect protein conformation?
You're increasing like the energy or the kinetic energy.
Yeah,
in the
because
proteins.
what is temperature when it comes to molecules? Vibration. Okay, what's holding the structure together? Weak forces between R groups. And if these are vibrating too much, it overcomes it. And likewise, pH. If you put in hydroxyl ions, it's going to interfere with ionic bonds. It's going to interfere with hydrogen bonds. You get denaturation. Okay. All right. So with denaturation, with heat shock, then all of a sudden, after that heat shock in the bacteria, then they notice that the genes coding for heat shock proteins, proteasomes, they increased, and the number of proteasomes in the cell increased, probably because of protein stress. Right? Okay. Okay, cytoskeleton. All throughout the cytoplasm are a network of fibrous proteins that are collectively called the cytoskeleton. And I'm going to, let's just jump a couple slides to 38, because I'd like to, first of all, I should put this slide earlier. What are these fibrous proteins, okay? So there's three different types of fibers. These are called microtubules. These are the largest ones. And these are hollow fibers made up of two different proteins that are globular. When I say globular, it means that the tertiary structure of these proteins is somewhat round, okay? And this is alpha, beta, tubulin. And these will form, so this is a hollow fiber, and we have 13 filaments, okay? So first of all, when these things line up, 13 filaments as a sheet, when they first start forming, then the edges come together to form a tube, all right? And then they grow from one end as two dimers, an alpha tubulin and a beta tubulin. These are two different proteins. They come from two different